In the nucleus of eukaryotes, chromosomal DNA is packaged into nucleosomes, which are repeating units of DNA wound around conserved proteins called histones. Two of the histones that mammals produce are H3A and H3B, which differ only slightly in amino acid sequence and are encoded by separate unlinked genes. H3 and H3B function during particular points in the cell cycle. Scientists hypothesize that histones H3A and H3B play roles in growth and development of diploid cells that divide to produce gametes. Okay. In order to investigate the relationship between the functions of H3A and H3B, scientists created two groups of mice. One group was heterozygous for deletion of the gene encoding H3A. So was this. Well, second group was heterozygous for deletion of the gene encoding H3B. So was that. Plus means functional allele, minus means allele with deletion, making the allele non-functional. A. Describe the expected genotype ratio of offspring from a cross between two mice that are heterozygous for a particular gene. So look, if they're heterozygous, let's just say they are capital H, small h, with capital H, small h. You get capital H, capital H, capital H, small h, capital H, small h, small h, small h. So when it comes to genotype, here we have 1, here we have 2, and here we have 1. So it's going to be 1 to 2 to 1 ratio. So we're going to say 1 is equal to homozygous dominant, 2 is equal to heterozygous, 1 is equal to homozygous recessive. The scientists performed a cross between H3 AA mice that were also H3B plus minus and determined the H3B genotype of the offspring. The scientists performed another cross between H3B plus mice that were also H3A plus minus and determined the H3A genotype. Okay, they then determined the mass of each of the offspring. In figure 1, mice that were homozygous for deletion in died. Okay, that's not good for them. Okay, so, oh yeah, they did die. Because if you have a double minus B, you die. So the homozygous for deletion. Wait, so if they were H3B double minus, they died during development. Yeah, so H3B genotype. If they were double minus, they would die. That's why they're going to have zero mass.
Oh my god. I think I had a mistake.
That's true.
I didn't read the H3B genotype. I just read the H3. Oh.
Okay, wait. So the average mass of 3-week-old H3AA mice. Is that an A? That's probably an A, right? Is that an A or a?
That's an A.
Okay. Identify a dependent variable in the first experiment in figure 1. So that is going to be what? The y-axis, right? So the average mass of the mice. Okay. So we're going to say the average mass of the mice. Double I. Justify why the scientists used the mass of wild type plus mice in the first experiment as a control. So this guy. And this guy. They're both plus. Yep. So let's see what plus means exactly.
I'm speaking randomly, I don't even know.
So plus means a functional allele. So they both have a functional allele, right?
It's like under the graph it's written.
Mm-hmm. Okay, so plus means functional. So the question says justify why the scientists used the mass of wild type plus as a control. Well, you want to just see, if there is any, like to compare the effects, right? So basically, this is to compare the effects of deletion of H3A/B alleles, okay, on the average. Is it the average mass of the mice to the control to see if the effects are significant or not, right? To see if the effects are significant or not. And the first experiment was only H3B. So H3B. Okay. Triple I. Justify why the scientists use mice of the same age for the first experiment as another control. Well, that way age is going to be eliminated as a possible, discrepancy. You could to eliminate age as a possible cause for the different results, to attribute the effects to H3A/B genotype deletions only. IV, based on the data in figure 1, describe the effect of losing one functional H3A allele, which means having 1 plus and 1 minus, compared to the effect of losing one functional having 1 plus and 1 minus in figure 1. So let's just compare them obviously to control, which is the wild type. So having a plus and a minus here results in a significant decrease of average mass, but having a plus and a minus here does not, because there's overlap, right? So the H3B plus minus genotype led to a statistically significant decrease in average mass of mice. I'm going to say compared to the wild type, right? But it's not the case for H3A plus minus genotype Okay. They said just describe due to error bar overlap with the wild type. Okay. The scientists then examined the structure of the gametes made by each of the offspring to assess the relationship between genotype and meiosis. Data for the analysis of sperm produced by H3A wild type and H3 double minus mice are shown in the figure 2. Okay, so no defects, like a lot more no defects in the case of AA, but a lot more tail defects in the case of double minus. CI, based on figure 2, describe the effect of the sperm structure of the H3A double minus. What do you think? So the H3AA, they had obviously more tail defects for the most part, and some head defects to a lesser extent. Compared to who? Compared to the H3AA genotype. Double plus, I mean. CII, assuming the scientists examined 200 H3A minus sperm, calculate the appropriate number of H3A minus sperm that had structural defects. So look, they are 15% no defect, right? So that means 85% is defected. So what's 85% of 200? That's going to be 170 basically.
Wait.
Yeah. You just have to draw the no defects to the thingy.
Why would you get 0.85?
Well, 15% no defect. How much is the defect from 100%? What do you think?
Sure.
Like 85%?
85% is different.
Yeah, because the whole thing is 100%.
Yeah.
Okay. D. I. State the null hypothesis for the effect of genotype in the second experiment. So the second experiment in figure 2. Null hypothesis means that the genotype has no effect on the sperm when it comes to having defects or not. Or basically you could say any defects or lack any defects in tail or head of sperm are not due to what? Are not due to the H3A genotype. I. Scientists claim that having two wild type H3A, that means having a plus and a plus, cannot restore the loss of the function caused by deletion in the H3B gene. Using data from figure 1 support the hypothesis. Let me bring the figure here. So we have to focus on figure A because that's the one with the H3A plus. And look, if you have H3A with a full deletion in the H3B, that means minus minus. All the mice died. So even though you had two functional plus for A, it could not save the mice because you had two missing B's basically. Okay. So yeah, that's how you support the claim. You have to, talk about having zero average mass. That means all the mice had died.